This is Team 17 on viral genome replication. Rotavirus is a contagious virus that causes inflammation of the stomach and intestines. It infects many children under the age of 5 and can cause fatal dehydration if not properly treated. Up until recently, the details of rotavirus biology have been limited by the inability to study the polymerases in the virus. On the left is an illustration of the symptoms of rotavirus, and on the right is a transmission electron micrograph of intact rotavirus particles.
This video will cover the role of RNA polymerase and the successful application of a RNA virus to infect a host cell and produce virion progeny. Viruses are dependent on host cell ribosomes for their protein formation and use the host polymerases for their own means, but also note that RNA viruses contain genes for RNA-dependent RNA polymerase in their genome. RNA viruses always encode to their own replication proteins because host cells cannot use RNA as a template for replication. For single-stranded RNA viruses, the process varies depending on whether the virus contains a positive or negative strand of RNA. For positive single-stranded RNA viruses, the viral genome is used to create the viral RNA polymerase and viral proteins needed to replicate the viral genome and synthesize the other molecules needed for proper reproduction. The RNA polymerase for negative ssRNA is contained within the capsid of the virus.
The research question that we present is how do RNA viruses replicate their genome within host cells? In a study by Biochem et al., a temperature-sensitive mutant of simian rotavirus strain SA11 was studied. The leucine to proline mutation is located at the N-terminus and was known to affect the VP1 polymerase. At 39 degrees Celsius, there is diminished growth and RNA synthesis compared to 31 degrees Celsius. The researchers wanted to investigate the mechanism behind the temperature-sensitive phenotype. They found that the leucine to proline mutation affects the ability of VP1 to localize to viroplasms, which are where RNA is synthesized. They fused VP1 with GFP to track its location in the cell. They also tested the ability of VP1 to synthesize RNA in vitro. They found that less RNA was synthesized at 39 degrees compared to 31 degrees. Data from Biochem et al. Showed the location of VP1 mutants fused to GFP in green and viroplasms in red. When the VP1 protein is localized with the viroplasm, the location is noted in orange. Note how at the permissive temperature on the left, the colocalization is seen. At the sensitive temperature on the right, there is no colocalization.
VP1 is an example of an RNA polymerase. RNA viruses use RNA polymerase to transcribe mRNA and replicate their genome. Without functional RNA polymerase, viruses cannot propagate. Research into the N-terminus mutation of VP1 helps us better understand the mechanism of viral propagation so we can develop drugs to kill viruses. Future research in this area will focus on further understanding of other viral propagation mechanisms and their ability to be manipulated. This graph illustrates understanding rotavirus as a compelling research question to treat other viruses.